We could envision that future of medicine may rely on giving a tiny hole on the cell wall to sneak new drugs. I am going to talk about electroporations, how microscopic holes could be beneficial in cancer therapy. It uses electricity to create temporary holes by overcoming natural cell defenses, and then functional molecules could enter and active inside a cell. In the case of non-thermal food processing, electroporation could enhance extraction of juice, sugar, oil, and health functional components. Lethal damage to the bacterial cells, keep fresh flavor, color, and nutrients, develop softer texture, and reduce processing time, for example, drying and frying. All these are possible due to the formation of tiny pores causing damage to the cellular membrane and their structure, thus enhancing mass flow. In the case of electroporation, pores are formed by reorganization of the lipid bilayer, hydrophobic tail, and hydrophilic ball and water molecules. These pores can be either resealed, that is reversible electroporation, or remained open, leading to cell death, that is irreversible electroporation. Therefore, we could develop temporary pores or permanent pores based on our requirement. Molecular components could enter or exit from the cell. How we could define electroporation? Electroporation is the process of temporary increase in permeability of a cell membrane by applying a high voltage electric pulse for a short duration. Therefore, there are three factors: high voltage electric field, electric pulse, and short duration. It is beyond a threshold, causing the cell membrane to develop transient pores. Now, what is the mechanism? In order to apply the electroporation, we need to understand its mechanisms or steps of formation of pores. Consider a cell at its normal state, that is initial state. The process takes 6 steps to form pores. These are applications of electric pulse, increase of transmembrane electric potential, pore formation, molecule entry into the cell, membrane resealing, or irreversible electroporation, that is permanent damage of the cell wall. I am going to explain these steps further. At the initial state, a cell could be resting at a membrane potential of around 100 mV. The cell membrane is a bilayer of phospholipids as shown in an earlier slide. A short, high voltage electric pulse is applied across the cell membrane. This causes transmembrane electric potential to increase. The external electric field adds to the internal potential, increasing the total electric potential difference across the membrane. This build-up potential sometimes reaching critical value of 0.5 to 1 volt, and it is the primary driver of pore formation. Induced transmembrane potential applied by electric field causes a rapid accumulation and redistribution of charged molecules. Ions on either side of the insulating cell membrane oriented along the field, and the next step is the pore formation. The increased potential causes the lipid bilayer to deform, creating temporary nanoscale pores that can span the membrane. Water molecules are pulled into the hydrophobic interior of the water-lipid interface of the membrane, facilitating the creation and opening of these pores. Pores are then stabilized. The water defects can expand and form a continuous water channel pores across the membrane. The adjacent polar head groups of the lipids are reoriented themselves to align the interior of this aqueous channel, thus stabilizing it into a hydrophilic pore. The diameter of these pores can range from 2 to several nanometers. Molecules can enter into the cell or exit from the cell through the channels created. For a short time, that pores are open. Then the increased membrane permeability allows molecules to enter the cell. It is driven by the electric field similar to electrophoresis. When the electric field is applied, the potential difference drives electrophoretic movement of charged molecules to enter through the newly formed pores. These types of molecules that could enter or exit depends on many factors. The size and duration of pores depend on the field strength and pulse duration. These also determine which molecules can enter. It is also important to know the existence and duration of pores to achieve desired purpose of electroporation. After the electric pulse is removed, the membrane typically reseals itself and trapping the molecules inside. This is called reversible electroporation and the cell survives. In the case when membrane rapidly reseals, then cells can recover and continue their normal functions. In addition, if entering molecules have a specific function to be performed inside the cell, then entering molecules could be active inside. We may also want to have irreversible electroporation. If the pulse is too strong or too long, the pores expand excessively and become permanent and unable to reseal, leading to permanent membrane damage and cell death through a process called irreversible electroporations. The pores expand excessively and become permanent. Then cells cannot restore their membrane integrity and die due to a loss of homeostasis. This is more relevant when we want to kill the cell, for example, pasteurization and sterilization. We need to know how to control electroporation and permeability. How to control the electroporation and cell wall permeability. Applied voltage, pulse duration, types of pulse, number of pulses, buffer composition, and cell types must be optimized to balance sufficient membrane permeability. Control is required for cell viability for successful applications in medicine such as gene transfer or electrochemotherapy. In the case of microbial cell destruction, the strength of the electric field must be high enough to complete disintegration of the cell wall. Thank you for watching this video until the end. It would be a great pleasure for me if you found this video interesting.